Should we just get started, Daniel? We've been running out of time, usually.
Yeah, sure.
Okay. So this is our practice session on August 15th. There's some background about Pre-Med Peers. If you're new, is that we're a nonprofit. You can contact us through Slack or email. If you have any questions, that's our email on the slide right there. So it's just you here today, Stephanie. Did you have any other questions before we get started?
Yeah, I actually do have a few questions. I missed, I think, the practice session last week, and I usually can't attend the classes live, so I was just wondering, when would most of the slides be posted? Because I keep up with the material. I just don't attend the classes live because I can't. But that's
so you're saying they're not up yet?
Yeah, there
I think,
were few of the classes.
Isn't the due date the Thursday of that when it's up, when the due date for all of the assignments to be uploaded, or videos, or am I wrong on that?
I'm not sure. Yeah, I didn't know that. I thought that was just I don't know. But usually it should be within the week. So I think you've done this before, but if you don't see the recording, you can just message us in Slack and remind whoever did the session to upload it.
Or just remind us all, because you might not know exactly who did the session.
Okay. All right. Thanks so much.
Is there anything else?
No.
All right. Great.
Yeah, and also, since it's just you, feel free to interrupt us or do anything, because it's all about you right now.
Yeah, no problem.
All right. Sounds good. So yeah, we have the 2 cars and psych-soc passages, which were assigned as homework, and then we're going to do CP and BBC Live. Did you get a chance to do the homework passages?
I already did them.
Okay, great. All right. The first one is the cars passage. Do you see the link to it in the chat, or do you need the link?
I don't need the link because I already did them. I have my notebook.
Okay, sounds good. Great. Okay, yeah. I think we can, Daniel, you can just go ahead to the next slide. Thank you. So the first question asks the passage would be most appropriate in a collection for, and then it's giving you a bunch of options on which type of collection would be most appropriate. So which one did you pick for this?
So I think initially I put choice A, but I understand why it's choice C now, because that one is the one that best fits the main idea.
Yeah, I also got strayed towards B, because it was talking about different types of people and then what they're stereotypically expected to do, but then I realized it was just a main idea question. So it wasn't really a political theory. It was talking about the cultural history, as in native Malays versus Chinese Malays versus Indian Malays. So it's talking about cultural identity and what people were essentially indoctrinated to believe they can do. But yeah, sounds good. And the next question says, prior to becoming British subjects, Malays were considered brilliant administrators and traders in Southeast Asia. What is the relevance of this consideration to the author's argument? So I think the first thing you should do in this question is remind yourself or in your head, what is the author's argument? So what did you identify as the author's argument that's relevant to this question?
Well, I chose the right answer. I chose C, but I mean, basically, the British, they kind of manipulated the image of the Malays, and to the point where they actually just, once they became independent, they weren't sure how to view themselves as.
Yeah, or more so it's to be more specific, they now view themselves as manual laborers only, whereas that, because that was the role that the British assigned to them over the course of their colonization period. But then if prior to that they didn't see the Malays didn't see themselves as just people who would be working in the countryside, then it does support the argument that the way they see themselves now post independence is because of what was done in the British colonial period. So yeah, sounds good. And then, which of the following Which of the following items of passage information provides the least support for the author's thesis? So we just said the author's thesis. So what did you pick for this one?
Yeah, so
Go ahead.
I picked B, but I know why it's wrong, because I think I chose the one that either one of the options that most support it. Sometimes I've noticed that I do that sometimes, where you click the one that actually supports it, even though they're asking for the one that doesn't. So I had to be more careful.
Okay, I see. Yeah. So the author's thesis is that the British manipulated like ideas about like a person, like a cultural group's purpose in that country. So then the fact A is like a very clear link to that thesis, so you would get rid of it immediately. B, I was a little bit iffy on, so I would come back to that. And then if we go to C, stereotyping was used as a tactic. That's for sure the same thing, because it's saying like they assign different roles to different cultural groups. So I would cross that off right away. And then D, I was between B and D then. So, but then if we think back to like the thesis being that the British manipulated these cultural like self-images, then D doesn't go into it. D is a fact that's stated in the passage, but the way the answer is explained about like why it's not D is because the fact that Malaysian politicians are attempting to redefine Malay identity was like an introduction into this idea of like why Malay identity is the way it is now, but it doesn't go along with the main idea of why of how the British manipulated Malay cultural identity.
Actually, when I did this, I thought they gave the correct answer as D. Maybe I was wrong.
Oh, sorry, you're right. Yeah, so the correct answer is D. I'll fix this. Yeah, so that's what I was saying. So the B does support the idea. So I didn't get to that yet. D doesn't support the idea because it's just introducing the concept of like why Malay identity is the way it is now. But the main idea is the fact is because like Malay identity is the way it is now because the British manipulated their self-image throughout the colonization period. So the way that B fits into that is because if the British saw that Malays were seen as deserving of protection, that's because they like pigeonholed Malay people into this idea of being like uneducated, like people who do labor in the countryside. So they need to be protected from the other like smarter culture groups who they cited as being like Indian Malays or like Chinese Malays. So that one, I can see where that's confusing, but does that make sense now? You're right, the answer is D.
Yeah, no, it makes sense. I just feel like if I spent another like minute on it, I probably would have recognized it, but because I was doing a little too fast, that's the reason why I got that one wrong.
Yeah, I think strike through really helps me out. Like, I think you really should only get stuck between B and D. So like visually, if you cross off A and C, I think it's super helpful. Okay, the next question. Oh, sorry. Also, Jennifer, you joined. Were you able to do these passages? Keep going. The author implies that Malays see themselves as essentially farmers and country peasants, primarily because of. What did you pick for this one? This also is just.
I put D.
Okay, perfect. Yeah. So the whole main idea was it's because the British kept repeating the idea that the Malays were farmers and country peasants. So it's not because of the demands of British rule. I think that's also like vague of like what the demands were. It's not like they were forced into doing it, but they were kind of like assigned roles and then they internalized that self-image and like purpose. So it's not the number of ethnic groups because it's because the British like prescribed these roles, not because there was like an extreme amount of ethnic groups and there's not enough roles for everyone. And then the location is never mentioned. So that one's good. And then the last question, passage information indicates that which of the following statements must be true?
I put B.
Okay, yep. So nothing is mentioned about a formal education system for A. For C, we don't know this. It doesn't mention it, like, but it sounds very possible, but it also could be not possible. So we just don't have enough information about this. And then lastly, for D, other British colonies are out of the scope of this passage, so you also wouldn't pick that. And then B, again, is the idea that the British kept repeating the fact that native Malays were a certain type of had to play certain types of roles, the other groups had to play certain types of roles. So they were manipulating what people thought of as their self-identity and purpose. So great. Thank you for participating. Do you have any concerns about that passage at all, or like just general card strategies before we move on?
No.
Okay, great.
Okay, so now we're going to the Psych-Soc passage. So if anybody wants to say what they got for this one.
I put what did I put? Roll strain.
Yeah. So this is definitely a very critical topic, just knowing the difference between role strain and role conflict. So I'd advise both of you just kind of nail this idea in your head. Just think conflict, like at least two different like things like conflicting with each other. So think conflict, multiple identities. So like, let's say you're a student, but you're also an athlete. That's kind of like the common idea. And then role strain is kind of within one thing, there's strain between those things. So like, you're an athlete, like you want to be the best athlete, but also like help your team as a whole. So, and then you probably both know this, but I always like to kind of make sure I know what each term is, even if I know the correct answer. So for role exit, I've seen that a couple of times in MCAT and prep material. Basically, role exit, it's kind of obvious. It's just exiting. So the classic idea is retirement, but it could also be like role exit, like once you graduate high school, that's a role exit from your role as being a high schooler. And then role enhancement, as I've wrote there, I've never seen it before, but I've kind of looked it up and it said that like one role has beneficial spillover effects on another role. But I've never seen that before, so I doubt that it would appear. Does anybody have an answer for this question?
Put B.
Yeah, so I wouldn't like memorize exactly what like Weber's ideal bureaucracy is. It probably would help you. I didn't memorize it, but I did know the 4 components of McDonaldization, which is kind of like, you can just think about it as like, what do you want in a fast food place? You want things to be efficient, you want them to be like calculable, predictable, and you want control over something. And then it's also important to know that a meritocracy is where people are selected based on their like ability. So I always think merit, they're selected on their merit, not like their social class or not anything else. So it's, you think bureaucracy, an ideal bureaucracy, yeah, you do want to like select people based on merit, but you do want like social stratifications and like specialized groups. But informal regulations, you definitely want like formal regulations as well. So that's where I selected that. So if you have any questions, feel free to ask. So we'll go on to the next one. Did anybody get this question on dreams?
I put C.
Yeah, so this is just something that you kind of have to memorize, the different theories of dreams. So basically, Freudian dream analysis, you think of Freud, that's kind of like, oh, there's these subconscious things. Think of the classic like iceberg. So basically his idea is like, oh, all of our dreams have these unconscious meanings in them. Threat simulation theory, that's kind of, you can kind of interpret what it is. It's basically just like kind of, you think of like the caveman brain, like, oh, we like stimulate threats. So for example, maybe you have a dream about like an elephant chasing you, and what would you do? Would you jump in a tree, then the elephant knocks it down and kills you or something? So that's kind of stimulating threats in the future. And then, so for example, for this, it's the activation synthesis theory, which is basically just like there's just random, not random, but the firings in your brain are like interpreted by like the like conscious part of your brain, and they kind of like messes up. So then that's their explanation for you why you get like weird and wacky dreams. So yeah. Any questions on that?
No.
Okay. Does anybody get an answer for this?
It's B.
Yeah, so basically this is pretty simple. This is just kind of classic dramaturgical theory by Goffman. So your backstage self is kind of, the person, if you're in a play or something, you're not on stage, you're not projecting. That's kind of like who you basically truly are when you're alone. But then the front stage self is like when you're surrounded by people. So maybe like when you're at a bar with friends, like you're very extroverted and you're friendly. But basically when you're home alone studying, this is who you are basically down to the core. So basically that's what it is. Alter casting and aligning actions, I tried to look those up. I've never seen them really in an MCAT context. So did anybody get an answer for this?
I put cerebellum.
Yeah. So basically this is just knowing a little bit of basic brain anatomy. So and like you don't need to know the details. So when I was studying, I just kind of put just basic snippets and associations. So for example, for this, I think pituitary gland, I think hormones. So that's not really, I mean, you can make a stretch with like acetylcholine and stuff, but that's not basically there. Then the medulla, I think respiratory system, so breathing. Cerebellum, that's coordination of like timing of muscle groups. So that could definitely be it. And then the nucleus, that's kind of just the pacemaker of the circadian rhythms. So that's more sleep-wake cycles. So it wouldn't really be with motor dysfunction. Okay, so yeah, you want to take over?
Should we, do you guys just want to read both passages together and then come back and do both passage questions, or do you want to do one at a time, come back, and then come back and take time to read the other one?
I guess you could do them. We could do them both at the same time. Like,
okay. So I put in the links in the chat. So I think they're pretty short. So maybe like 15 minutes. If you need more time, we could give you more. Does that sound good? Okay, great. So yeah, Daniel, do you have anything else to add?
No, I think that works.
All right. So we'll see you guys. If you do get done early, let us know in the chat so we can come back earlier too. Are you guys about done reading it, or do you need more time? Or I guess it's just you now, Stephanie.
Can you give me another 2 more minutes?
Yeah, just let us know when you're done. Thanks.
All right, so we can go through the rest.
All right, sounds good. Do you have any questions on the passage itself?
Not in particular. I mean, I already know which ones I got wrong.
Okay.
Yeah.
Yeah. All right. So, in a mixture of lanesterol and mevalolactone, which IR stretch would not be detected in a spectrum analysis? So, Daniel, if you could hit the next 2 slides, like 2 more images should pop up. Yeah. So these are your compounds. So if you have a mixture of them, you're going to have all the functional groups between them. So you're going to have the OH, the alkane, the lactone, which is like the cyclic ester, and double bonds also in the lanesterol. So those are the bands that you're going to see. So I would just go one by one through the option choices and identify those. So if you could hit the spacebar one more time, then yeah, these are your stretches. So if we look at the first one, broad 3200 to 3600, which one is that?
That's the OH group, right?
Yep. So we cross that off. Next is your narrow 2800 to 3000. Which one is that?
That was the one I picked, and that was the one that was wrong.
Okay.
So,
yeah, just go ahead and look at the chart now. And then
I have to zoom in.
Can you see it or just?
Yeah, I'm just looking through right now. It says it's between 2800. Oh, so maybe it's an alkene.
Just a regular alkane.
Alkene, yeah, I see that now. Yeah, I just never had them really memorized. So
yeah, I can't actually remember. This one caught me by surprise. I don't know if we had to know IR stretches like this specifically. Obviously, everyone should know like the OH one. What do you think, Daniel?
Yeah, this is one of those things where I think like if you really want to get like a high score, you should memorize it. But as long as you know like the very basic like OH, carboxyl groups, as long as you know the basics, you should get still like a very good score. Kind of like I said about the roll exit, and then see now I forget what it was, the like roll enrichment or something like that. Like if you want to get like a really high score, that's what you got to know. But you don't need to get like an insanely high score to get into med school. So if you have time, I would memorize them. But if not, just focus on the main ones.
Okay.
Yeah, because you probably knew C, right? I would say that's one you should know as well. That's your carbonyl.
Yeah, that's the carbonyl one. Okay. That's 1700. That's the one I have memorized. And then I guess, so then what would be D? then
it looks a little bit different on this graph, so that's also something to keep in mind. But it's on this graph, it's actually between more like 33 and 3400. This in the answer explanation, they said this was the amine. So like the blue one on the very left.
That kind of makes sense, because I remember I was watching the videos for the class that they said that the carboxylic acid is like broad.
Yep.
And then the amine ones is more narrow. So it has the same number. So
yeah, that's a good way to differentiate it. Yeah. So the amine one is the one that's not in any of these two compounds. So that's your answer then. So then next question was, which of the following can isoterpene least likely to be made into? Did you get this one?
Yeah, I got it. It's glycerol, because the isoprene, they make basically, if you look at the pictures, they make cholesterol, and I knew they made terpenes, and estrogen's a cholesterol
perfect
hormone. So
yeah. And then how many carbons and phosphorus atoms are found in a molecule of FPP? So this one is like knowing how to approach this question. Did you find?
I actually did get it right. It took me a little bit of time, but I just kind of counted the GPP carbons, and then I counted the IPP carbons, and then I got up to 15, and I was like, okay, I hope this is right.
And what about the phosphates? Because you would think that if you have 2 phosphates in IPP and then 2 phosphates in GPP, it would be 4 phosphates.
See, that's what I was thinking too, but I just went with the number of carbons. Maybe because the 2 phosphates, maybe they act as like leaving groups.
Exactly. Yeah, and you don't have to guess that. It's given to you in the passage. Oh, sorry, I can't find my window right now. Just give me 1 second for me to pull it up. So if you look in the second paragraph, it says in the middle of the second paragraph, GPP is combined with IPP and creates FPP and 2-phosphate units. So if those 2 things are coming together to form GPP and 2-phosphate units, it means, like you said, the 2-phosphate units are leaving. It's about like just like getting those details right. Obviously, in some cases like this, you can get away with not reading it, but I think that's actually like something really easy that a lot of people miss, is like reading like 2 words that can basically give the answer away. Yeah, so that one's good. And then a researcher uses thin layer chromatography on silica to monitor the progress of the conversion of mevalonate into R-mevalonate diphosphate. Does the product have a higher or lower RF value than the starting material?
Yeah, so it's D, because the silica, and this is something I always get confused, and I almost did it to myself too. I always think the silica's nonpolar, but it's polar.
Yeah.
And then the multiphase is nonpolar, and then you can kind of figure it out, because RF means like the distance that's traveled.
Yep, perfect. Yeah, so I just think of it like you said, the phosphate makes it more polar. So then polar is going to stick to polar. So if we know silica is polar, the mevalonate 5-phosphate is going to stick to the silica more, have a lower RF, like you said. So yeah, that's perfect. And then this is the last question, but this is probably the hardest question. In an alternative pathway to generate lenesterol, a prosterol cation can be enzymatically converted using the following mechanism in reaction 1. Which carbon contains the hydrogen that is enzymatically removed to begin the conversion?
So I actually did get this right.
Nice.
It took me some time. Yeah. I don't know. I guess like I kind of converted whatever the chair was to the normal, like, I forgot what the word is, like
I don't think.
And I somehow got to 9 instead of 8.
Yeah, I don't even think you have to convert like both to chair and all of that. It's just matching like the bottom diagram to like the one on the top right. So Daniel, if you could click 2 times. Yeah, so this is how I matched it up. And honestly, like this is so not content or like any knowledge. It's just like being able to like look at a diagram and like match things up. But a lot of the orgo questions are like this. Like if they give you a reaction sequence, they'll be like which you kind of just have to like visually be able to track through the different regions, like what's happening, which carbon is losing which or whatever. So what I did was like, so 10, if you look on the bottom figure, is like between the left 2 rings, right? So then that's how I matched it up to like blue to blue. And then 9 and 8, we can tell those are the connecting pieces between the second ring from the left and the third ring from the left. So then I matched up 9 as like the top connector between those 2 rings, and then 8 as the bottom connector between those 2 rings. And that's basically how I did it.
Yeah, that's pretty much kind of how I thought about it too. I just kind of drew a little picture to help me out.
Okay, yeah, that works. All right.
Okay. So sorry to jump right into it, but did you have any questions on the bio passage?
I was like, I'll write on it. I got like only 2 right. No, I got this question wrong too. I don't know.
Okay.
Wrong.
Yeah, so basically for this one, it's just saying that like so the enzyme activity is going to be like a little lower as you saw in the graph. And so if there's just, this was kind of like just following a pathway. So the way I thought about it is that like if the succinate dehydrogenase activity is lower, there's going to be less electron train transport activity and then just less ATP. And then so this was kind of the answers weren't really like scientific in a sense. It's just kind of saying that, okay, what is going to happen if we have less ATP? And you can kind of go through them. And for me, for this question, I wasn't too sure about the last 3, but I knew that like motility, you need ATP to kind of power that tail. So that's kind of my logic with this question.
Yeah, so I went for B, but then I could kind of understand how, because maybe in my mind I maybe misinterpreted what standard reduction potential meant for FAD, because I thought maybe there would be less FAD like present because it's destroyed, because it's bad. But I guess like I don't like, what does standard reduction potential mean? Like the number they give you or something?
Yeah, so from what I'm pretty sure from what I remember that standard reduction potential, that's just basically like an intrinsic property like of certain chemicals to be reduced. So for example, like I think later along in this PowerPoint, I have kind of like a chart that shows it, but it just kind of shows like the path of an electron from like the carbon, like a based form like glucose, all the way down to like water and oxygen. And it's just basically like the affinity for it to want to either grab or donate an electron. So I'm trying to think of it right now, but from what I remember, it's just kind of like an intrinsic property. So you could change it maybe by altering the molecule, or maybe altering its environment. But yeah.
Okay. All right.
Oh, and this is just something I also added that, because this is one of those like small facts that like I didn't really see reading like the Princeton review books or anywhere, but it appeared a couple times in like UWorld questions. But succinate dehydrogenase, which is also known as complex 2, it's part of the electron transport chain and the citric acid cycle. So I think I just put this there just as like a little bit of tidbit for information, because I remember I got a good amount of questions on the AAMC practice materials. Like there are kind of questions on that. So just a little fun fact to know. So did you get this question?
Yeah, I got this one.
Awesome. Yeah, so basically, like as you probably know that we have this bacteria growth phase. So we have like the lag phase. That's kind of like when the bacteria are growing, they're kind of adapting to the situation. And then all of a sudden it's the log phase. That's when they kind of exponentially grow, because as you see, it's like a log on the axis. And then depending on whatever the environment is, like then there's the stationary phase where they're just kind of chilling. And then ultimately, if it's like an agar plate or something where there's a set amount of nutrients, ultimately the colonies are going to die because they have no more nutrients. So yeah, basically that like the more you put of this toxin or chemical, you're gonna have longer lag phases.
So basically slower growth. Like,
Yeah, yeah. Did you get this?
Yeah.
Yeah, so basically this is just kind of knowing the different metabolic pathways. One of the critical things besides amino acids to just kind of memorize for the MCAT. So yeah. Did you have any questions on this in particular or?
No, actually.
Okay.
I have pretty good knowledge of biochem because I did a master's program. So.
Oh, really? That's awesome. Yeah. Did you get this question?
No. Yeah.
Yeah, so this one I kind of just went through it based on the experimental protocol, which characteristic explains their decision to use them. So basically, so I just kind of went through the answers because this is kind of one where it's not like a scientific answer, it's just kind of more using your outside, just general logic. So for what answer, did you choose an answer or?
I actually chose D.
D? Yeah, so the thing with D is that it's definitely true. It definitely could be true, but just in terms of like scientific experiments, A is kind of the better answer. Just basically because in the paragraph, and this is kind of like how previously there's like these little like hints, and it's annoying because sometimes I found for like the bio passages, you don't have time to completely read through it. Like sometimes I would just skim through it to get the main idea. But then sometimes there's also like these little bits of information. So like I highlighted here that the bacteria was grown at 65 degrees Celsius to heat kill any other organisms. So like they specifically mention that like, well, they don't specifically mention, but it's kind of implied that like the other organisms died, whereas this organism, it has high heat tolerance. So it's basically just their way to ensure that bacteria X is the only species there. So while like D could also be true. See, I get your reasoning though. It's kind of like this is the best answer, but just from an
from the main idea. But I can also see, because I've done practice before. Now, sometimes when I don't do it on a time limit, let's just say, like I might have been able to catch that because it makes sense. I think because like it's at a high temperature for bacteria. I don't know what temperature they grow at, but it did say that it heat killed any other organisms. So maybe A would be the better answer.
Yeah. And I don't remember exactly, but I don't remember seeing anything talking about like it being like a common target. But like, again, it's kind of like that you can also think of with your background information. But yeah, it's kind of, I think for me, the keyword here was experimental protocol. So I thought about like what makes this bacteria unique? Like why would they want this in particular? Like I don't know too much about DDT, but I would imagine that many bacteria are affected by this because it's like a powerful toxin. So yeah. Okay. I thought there were 5 questions, but I guess there were 4. Yeah. Thank you for joining us. It's been awesome. Do you have any further questions? Anybody here or?
No, but so I only took like some half lengths, like 2, maybe the last one I took was like 3 weeks ago, and I want to try and take the test in like December, January. So like, would you? Because I have to finish one class in my master's program. Like, would I start like doing like UWorld questions like for like whatever how much I paid for, which was like 3 months, like around this time now? Because I've been doing a lot of practice from like all the free stuff.
Yeah. So take everybody's advice with a grain of salt because like what worked for me might not work for others. Generally, so August, September, October, November, December.
Oh, I'm taking a lot of time because this is a retake.
Yeah, no, that's perfect. I'm just kind of calculating it, like because when I did UWorld, I kind of pushed it off to the last minute, so it ended up I only did 2 months of UWorld, and I wasn't able to get through all of the questions. I got through like 3/4 of the questions, So it should, if you're like dedicated, 3 months should be enough for UWorld. My advice to you would be kind of budget 3 months for UWorld, and then 1 month for all of the AAMC materials if you want to purchase all of them, which I would recommend.
Oh yeah, I'm gonna purchase them probably by the end of this month.
Yeah, I think it's like $300, but it's definitely worth it. It should take you about a month to get through all of that, including the 4 full-length exams. So that's 4 months right there. So you have a little bit of time to do like some more content review, or whatever. Like what I did was I just skimmed over the Princeton Review content books and did a couple questions. I didn't get through like not even like a tenth of the books, but if you have extra time, you could do that, or you could get a head start on UWorld questions, because as you probably know, like the UWorld questions, they're absolutely amazing. Honestly, I don't even, as long as you have a somewhat strong background, and you seem like you do, I don't even think you need much content review, because
Yeah, no, the only thing I've needed the reason why I did this like course was for like chem and physics and orgo. Psych, like you could learn on your own. Bio, I took like a really intensive master's program. So, and they told us it would help us for the MCAT, which I mean, it kind of did. But I was just asking because like I have to score pretty well, like at least above a 510 to make it look worth it for me, because I did very well in my master's program. So like, and my MCAT that I took like 3 years ago was like not like a 502 or 503. So that's why I was just asking. But thank you for the advice.
Yeah, you definitely seem like you're in a good boat. Definitely start UWorld earlier than later. You'll see this advice a lot, I find, is that focus more on questions and full-length exams versus like content review. A lot of people make that mistake where they think like, oh, I don't know this topic enough. Let me spend like a month reviewing it. And then,
yeah, I get that, because I haven't really been doing that. I've been practicing. I've just been using free stuff.
Yeah, because you'll find that it's for like physics and orgo, it's not a lot of questions. Like it's like, like wavelength spectrum, like there's not a lot of kinematics. So it's kind of like as you go through the UWorld questions, if you get a question wrong and you're like, I don't know this content at all, then you can review that specific topic and it saves you time.
Okay, well, thank you so much for the advice. Maybe I'll come back here for another practice session.
Yeah, thank you so much.
All right, you guys have a great one. Bye.
You too. Have a good day.
Jennifer, are you here at all? If not, we're just going to end it in a minute. I'm going to stop recording.